our formulas 421 medical hematology 1. Okay, basically in this course, this section of the course, we will be looking at hemostasis and thrombosis, the basics of hemostasis and thrombosis. Okay, we are going to look at every aspect of hemostasis and thrombosis. Our course outline has two parts to it. We have the introduction to hemostasis. Our part B, the part B of this course will entail studying some disorders of hemostasis. So for the part A of this course, we are going to look at hemostasis and overview. We are going to look at an overview of hemostasis, breaking it down into primary hemostasis, secondary hemostasis, the fibrinolytic system. Then we are going to look at thrombosis and and we are going to look at thrombosis and some thrombotic disorders. So taking the first aspect of this part A, hemostasis and overview, we are going to do an overview of hemostasis. Okay. We have primary hemostasis, have secondary hemostasis, we have fibrinolytic system, and we have the thrombosis and thrombotic disorders. So our learning objective includes that at the end of this lecture, you should be able to define what hemostasis is, okay? Be able to also enumerate some of the laboratory tests involved in hemostatic function, understand the details, the underlying mechanisms of bleeding and thrombotic disorder, be able to classify and describe clinical manifestations of bleeding and clotting disorder. Okay, you should also be able to describe the role of endothelial cells in blood vessel integrity and hemostasis. You'll be able to list the natural anticoagulants that prevent blood clotting and their mechanism of actions. The fibrinolytic system, you should be able to explain the process of breaking down blood clots. So as a way of definition of terms, okay, the hemostasis refers to all the physiological response that prevents or arrests blood loss following vascular injury. Okay, so by this definition, we are trying to say that the processes, the physiological processes by which the body tries to prevent blood from gushing out or leaving the vascular system is what we refer to as hemostatic mechanism. Okay, it is a complex system, okay, and it involves interactions between the cellular and the plasmic blood components with the vessel wall to ensure that blood flow in a fluid state. So what it also tries to imply is that as you are seated on your seat listening to this lecture without any cut or wound in your system, basically your blood don't need to clot. Your blood should be allowed to be flowing through the vascular system in and out of the heart, pumping blood, getting to all the organs of the body. So it is the hemostatic system that ensures also that the blood remains in fluid state as it circulates, okay, inside the body. This hemostatic system is also responsible in preventing blood loss in case where somebody encounters cuts in the body. So this is the definition or explanation for the hemostasis system. It ensures that blood remains in the fluid state while circulating in the vascular system. It also ensures that it also involves the physiological response by the body to prevent the loss of blood in case of vascular injury. Okay, then another terminology we have is thrombosis. Okay, thrombosis define pathologic formation of blood clots in the blood vessels. So thrombosis is trying to define a situation or a condition where, just as I said earlier, as you're seated on your desk listening to this lecture, you don't have any wound, you don't really need your blood to clot because you are not really losing blood per se. So your blood doesn't need to clot while you don't have wound. But there are some situations, we call them pathologic formation of blood from blood clot. Pathologic in the sense that that clotting, it's not needed at that particular time. Take for example, because of the deposition of fats in the blood vessel walls and narrowing of the blood vessel, we may have some plaques formation, blood clot forming inside the blood vessels, and this can obstruct blood flow into our some of our vital organs, cutting off these organs from the supply of blood. And when any organ of the body or part of the body is cut from getting supply of blood, that organ will definitely die off. So these two terms, we need to understand it very well. Hemostasis involves all physiological involves maintaining the blood in fluid states as it remains circulation. All the physiological uh, uh, all the physiological properties of the blood and even the endothelial cells, as we'll be learning, that contributes to the maintenance of blood in fluid state, and also all the physiological response from the blood system, the components and endothelial cells that makes them to be able to stop or arrest blood from flowing out of the vascular system in the course of injury, explains the term hemostasis. While thrombosis, we say it defines pathologic formation of blood clots in the blood vessels. Right. So the relevance of hemostasis. Why is hemostasis important? We say that the hemostasis is very important in the system because it helps to maintain in the blood in a fluid state while it remains circulating within the vascular system. Okay. It also maintains integrity of the vessel wall. This is in a situation where somebody maybe had an injury or there was a cut, and after um, the the physiological response by the hemostatic system to stop the blood, okay, it also ensures that the blood vessel wall, integrity of the blood vessel is maintained, okay, because building up the blood clot to stop bleeding does not go on indefinitely. Once the blood the bleeding has been stopped, the fibrinolytic system sets in, the clot has to be resolved so that the blood vessel, the patency of the blood vessel will be maintained, the integrity of the blood vessel, sorry, will be maintained. Okay, it also it's important because to arrest bleeding at site of injury or blood loss by formation of hemostatic plugs. Then eventual removal of plug when healing is complete. This is why the hemostatic system is very, very important in our body. Okay, so as a way of introduction, we are going to look at the role of blood vessels in hemostasis. Okay, you know that the blood we are talking about flows through the vascular system to all and from all the parts of the body. The vascular system consists of, we have the capillaries, the arteries, and the veins. Okay. So blood normally flows within blood vessels whose physical capabilities include contraction, that is narrowing, dilation, widening, which are controlled by the smooth muscles of the vessel media, that is the endothelial cells. Okay. Vasoconstriction and vasodilation provide the means for control of blood flow rates and blood pressure. Substances released from the endothelial cells and subendothelial smooth muscles also contribute to normal blood flow and prevent abnormal formation of clots. All these statements in this introduction will explain it as we go on to describe the various functions of some of these cells and the endothelial cells also. So the layers, when you look at the blood vessel through the blood in our system flows, they are made up of 3 layers: the intima, the tunica intima, the media, and then the adventitia. Okay, the intima, the endothelial cells and the subendothelial cells consist of the endothelial cells and the subendothelium. The tunica media made up of smooth muscles, the muscles that cause the contraction, and the extracellular matrix. Tunica adventitia, there we have the fibroblasts. You can see it in this diagram. You get the fibroblast there. Okay. You get the fibroblasts. You get the cellular matrix, loose connective tissue. You can see the tunica adventitia, tunica media, and tunica intima. So the endothelial cells is a one cell, single cell thick. So that is a simple structure of the endothelial cells. Okay. So the endothelium versus the subendothelium, that's the endothelial cells. These endothelial cells line the vessels. The innermost layer of the vessels are lined by this one cell thick endothelial cells. They line the inner vessels of the blood vessel wall of the capillaries, the arteries, and the veins. Okay, they are thromboresistance in nature. Okay, they resist the formation of platelet plugs. Okay, which is understandable because inside the lumen of the blood vessel, where there is no cut, there is no injury, you don't really need the blood to clot. So the endothelial cells by nature are made to be thromboresistance because of some substances they express or secrete, such as thrombomodulin and heparin sulfate, to keep the inappropriate thrombi from forming. They also release tissue plasminogen activator and urokinase in the presence of thrombin, shut off the coagulation cascade in the presence of thrombin. Okay. Then the subendothelium, immediately after the endothelial cells, we have the structure we call the subendothelium. Immediately after the endothelial cells in the vessel wall, beneath the endothelium, are thrombogenic in nature. You see, these ones are thrombogenic. That is, they enhance the thrombosis. That's why they are saying the subendothelium is thrombogenic. They enhance thrombosis, the formation of plaques. Why? Because they express some substances inside of them, like the von Willebrand factor. Will be learning how it plays a role in this mechanism, as the collagen is contained on the subendothelium, and also tissue factor to kick off coagulation cascade. Because what it implies is immediately the subendothelium, if the subendothelium is exposed, that means the endothelial cells has been injured. There's a cut that have gone inside to now expose the subendothelium. When the subendothelium is exposed, its contents that it express are also released, And some of these contents that it contains enhances the formation of uh, uh, plugs to stop whatever have caused the triggered of the bleeding. So that's it. So we're going to look at now the hemostatic mechanisms, just like in the earlier slide we have checked. We have the primary hemostasis, we have the secondary hemostasis, we have the fibrinolytic system. Okay, so the hemostatic mechanism, the process by which the blood is maintained in the fluid state, blood clots if there's an injury, and also the blood, there's a resolution of the clots, are divided into some three major steps. We categorized into primary hemostasis, secondary hemostasis, and the fibrinolytic system. So today we're going to look at the primary hemostasis in details, and it's very, very important that you understand this primary hemostasis very, very well. So in primary hemostasis, okay, the first step in primary hemostasis, the first step is the transient constriction of damaged vessels mediated by reflex neural stimulation and endothelin released from the endothelial cells. So immediately there's a cut, what the blood vessels will do is it's a reflex. They will try to like constrict constrict. Some of us have seen where they killed their fowl. You can see some of the blood vessels trying to, as if they are checking out. So it try to constrict. That constriction is to narrow the lumen through which the blood is gushing out. So the blood vessels, the first thing in primary hemostasis, the first primary thing the blood vessel does as to stop bleeding is this transient constriction because of some reflex neural stimulation and also the endothelin that is released from the endothelial cells. The second step involves, we're going to also see this diagrammatically, but let's explain it here first. So the second step involves platelet adhesion to the surface of disrupted vessel. We're going to look at it diagrammatically, and you see the components that are also involved in this. Okay, for the second step to occur, we say that von Willebrand factor binds exposed subendothelial collagen. You know, we mentioned that if the subendothelium contains some substances like von Willebrand factor, collagen, okay, and if this subendothelia is being exposed, okay, the von Willebrand factor will bind exposed subendothelial collagen. Okay, the von Willebrand factor is derived from the Weber palette bodies of the endothelial cells and alpha granules of platelets, because during this injury all these cells are also affected. Then the platelet binds von Willebrand factors using the glycoprotein 1b receptor. Okay, now the third step in primary hemostasis involves platelet degranulations. Okay, the adhesion of platelets, okay, to the von Willebrand factor induces shape change in platelets. It makes the platelet to have a change in their shape. This enhances the degranulation, that is the release of granules. These are multiple mediators, including the ADP, the dense granules coming from the dense granules. That is, it will release ADP. The ADP is stored in the dense granules of the platelets, and the ADP, what it does is to promote exposure of glycoprotein 2b3a receptors on platelets. Okay, the degranulation also releases thromboxane A2 that promotes platelet aggregation. So the activation of platelets causes the platelet release of the dense granules of platelets, which contains ADP. Then when the ADP is released, it promotes the exposure of more receptors on another receptor on the platelet, that is glycoprotein 2b 3a receptor on platelets. Okay, then we'll see the function of this new receptor that comes up as we go on, and also thromboxane A2 that is also being released as a result of platelet activation. And the function of the thromboxane A2 is to promote platelet aggregation. Then the fourth step in primary hemostasis is platelet aggregation. Okay, the first one we talked about adhesion. Okay, we talked about platelet adhesion. If we go back, we talked about platelet adhesion in step 2. Okay, adhesion, platelet binds von Willebrand factor using glycoprotein 1b. This is platelet adhesion, and this platelet adhesion causes platelets to be activated and release some of these dense molecule substances, ADP. Okay, so after this activation of platelets and also the expression of the second receptor on platelets, that is glycoprotein 2b 3a receptor, the process that is platelet aggregation now comes into play. Now, platelet aggregation at site of injury. Platelet aggregates at site of injury via this glycoprotein 2b 3a using fibrinogen as a linking molecule. This results in the formation of platelet plug. So the platelets that have been activated after the adhesion expresses a second receptor, that is the glycoprotein 2b 3a. Then using fibrinogen is a soluble protein in the plasma. Using fibrinogen as a linking molecule, these platelets link each other, forming what we call the platelet plug. This results in the formation of platelet plug. Okay, right. So this is a diagram that's tried to explain the process of basically the primary hemostasis. So when you look at this diagram, you have see the hemostatic plug formation, okay, in response to injury. In response to injury, we're talking about in response to injury, okay. So we have the first initial response, we said vessel constrictions. After that, the formation of an unstable platelet plug. Why do we call it unstable? Because that initial platelet plug that was formed using fibrinogen as a linking molecule, linking the platelets together through their glycoprotein 2b3a receptors, okay, that initial plugging are unstable. If the force of blood flow inside the blood vessel is high, it can dislodge that clot and it will not be able to really prevent blood from flowing out. So that initial platelet plug that ended primary hemostasis, because that platelet plug formation is where primary hemostatic mechanism uh, ends. Primary hemostasis ends with the formation of platelet plugs, that is platelet aggregation. And we say that those platelet aggregates, that they are unstable and can easily be pushed away or dislodged by the blood flowing inside the blood vessels. So there is need for that platelet plugs, that primary hemostasis, to be solidified. And that is when the process of uh, secondary hemostasis comes into play. The secondary hemostasis essentially leads to the stabilization of the plug with fibrin. Okay, so the secondary hemostasis is essentially to convert that fibrinogen into fibrin, which is more stable than the platelet block, and that is the area for the secondary hemostasis. Then the dissolution of clots and vessel repair. This is where the process of fibrinolysis comes into play. Okay. So looking at this is a diagrammatic explanation so that you'll be able to really understand the process of primary hemostasis very well. So this is an injured blood vessel. You can see the endothelial cells, the one cell T. You can see how it shifted one side. It's been injured. Okay, so once that it is done, the subendothelium has been exposed. We can see the von Willebrand factor. Okay, linking to the platelets with the glycoprotein 1b receptors, or the platelets can also use its glycoprotein 1 to also link to the collagen in the subendothelium. We say that the subendothelium contains both the von Willebrand factor and the collagen. Either of this can happen. Okay, then with the adhesion, this process we call it the adhesion of platelets. Okay, platelets adhere to the subendothelia through von Willebrand factor or through the collagen, or by the collagen using their glycoprotein 1b. Here is glycoprotein 1a to either link to the collagen, or glycoprotein 1b to link to von Willebrand factor. That is platelet adhesion. Then we say that this platelet adhesion results in platelets what? Activation. When platelets adhere, they are activated. And when they are activated, they begin to release their granule contents. They release ADP and what? Thromboxane. Okay. So when that happens, just like I was trying to explain, that the we say that the excuse me okay, we say that the thromboxane, the ADP and the thromboxane that are being released, and that will result in platelets expressing another receptor. You can see it here, glycoprotein 2b3a receptor. Okay, when platelet is being activated by the adhesion, it expresses another receptor we call the glycoprotein 2b3a. Okay, and this receptor binds it with each other, each platelet, using this fibrinogen. You can see it here, fibrinogen, as a linking molecule. Okay, so platelets, by expressing this second receptor, binds to each other using fibrinogen. You can see the fibrinogen here as a linking molecule, forming platelet aggregation. Okay, so activated platelet localize coagulation to the site of injury. So these activated platelets makes it possible for the process of coagulation to be centrally localized. Okay, it's not something that will spread throughout the vascular system causing coagulation everywhere. No, there's a system that makes sure that at that site where the injury occurs is the site where the coagulation mechanism is taking place. It doesn't have to spread throughout the vascular system, otherwise it will be another disease condition. So the activated platelet localize coagulation to the site of injury. Okay, so, and we say that the formation of platelet aggregates, that formation of platelet aggregation, marks the end of uh, primary hemostasis. So next we'll be looking at secondary hemostatic mechanism. We'll be discussing the process of converting the fibrinogen. As you are seeing it here, this is the blood coagulation cascade. We'll be looking at the models, because we have the cell-based models, we have the cascade models, the models that describe this coagulation cascade, secondary coagulation mechanism. We are going to discuss secondary coagulation mechanism using the two base models, the cascade models and the cell-based models, that its ultimate aim is to convert that fibrinogen that was used as a linking molecule in primary hemostasis, to convert it to fibrin and form a more stable fibrin to stabilize the plug, so that it will not be dislodged from as the blood flows. So I actually want to make this an introductory lecture so that others, because don't join class on time, so that they will know that we have actually started. So this